Welcome back to the deep dive. Today, we are sinking our shovels deep into the soil to explore the silent masters of the earth, kingdom fungi.
Right.
And, these organisms are so much more than just the mushrooms we see after a rain.
Oh, absolutely. They're the planet's essential decomposers, nutrient recyclers. I mean, they're ancient ecosystem engineers. And yet most of them are completely hidden from view.
That's what's so
It
fascinating.
is. And our mission today is to really systematically explore their world. We'll be looking at 5 core themes.
Okay, what have we got?
First, how their unique structure lets them feed externally. Then the strategy behind their spore-based life cycles.
Their evolution, too. I heard they have a surprising kinship with animals.
It's a very surprising kinship. Then we'll cover the sheer diversity of their modern lineages, and finally, their indispensable roles, both good and bad.
From medicine to, massive crop loss.
Exactly. We're going to walk through all the mechanisms that make them tick and hopefully help you visualize these really complex biological processes.
So let's start with that fundamental question. How exactly does an organism that can't move or, ingest food, how does it manage to eat?
That is the perfect place to start. And the answer is in their method of nutrition. Fungi are heterotrophs.
Meaning they get nutrients from other sources?
Correct. But they don't ingest food like an animal does. Instead, they feed entirely by absorption. They're the ultimate external processes.
So if they can't bring the food inside, they have to bring the inside out.
That's a great way to put it. They secrete these incredibly potent hydrolytic enzymes into their surroundings.
And these enzymes are just breaking everything down.
Think of them as biological chainsaws. They break down massive, complex molecules, things like wood, lignin, even jet fuel, into tiny compounds.
Wow.
And once they're broken down, the fungi just absorb those simple molecules directly across their cell walls.
Which explains their incredible ecological range.
It does. It lets them be the great decomposers, breaking down non-living matter, or they can be parasites, absorbing nutrients from living hosts.
Which can cause disease.
Often, yes. Or they can be mutualists, where they give something back, like helping termites digest wood.
And this unique way of feeding, it dictates their whole body plan, right?
It has to. To absorb nutrients efficiently, you have to maximize your surface area. And they do this through a filamentous structure.
Okay, so the basic unit is the hypha.
The hypha, yes, a tiny tubular filament. And when you have an interwoven mass of these hyphae, that forms the main feeding body, the mycelium.
And this structure is, it's genius from an engineering perspective.
It really is. That enormous surface-to-volume ratio.
Means the mycelium can just, completely mine an entire substrate for nutrients.
The scale is mind-boggling. People have found up to 1 kilometer of hyphae woven into a single cubic centimeter of rich soil.
1 kilometer. That's incredible.
So you have to think of it like a giant sponge just spread out through the entire environment. But this creates a problem.
A physics problem.
A physics problem, yes. Pulling in all those concentrated nutrients creates massive osmotic pressure. The rapid influx of water threatens to just burst the cell.
Which is where the cell wall comes in. It's reinforced by kin.
Exactly. Kinin. That polysaccharide provides a flexible but incredibly tough scaffolding.
So without that rigid protective wall,
The whole system would fail. The hyphae would rupture immediately. Tins is what makes the whole absorptive lifestyle possible on land.
and within that wall, there are different ways the cells are organized.
Right. You have what are called septate hyphae, which have these cross walls, the septa, to divide the cells.
But they have pores.
They do, big enough for organelles and even nuclei to flow through. And then you have the coenocytic fungi.
And they just don't have the walls.
They forego the septa entirely. You literally get one continuous cytoplasmic mass with thousands of nuclei. It's like a giant multinucleate feeding machine.
Wild. Now let's zoom back out to those mutualists. We have to talk about mycorrhizae, the fungus roots.
Yes. You could argue they fundamentally reshaped life on land. They are, for many plants, non-negotiable,
So the fungus helps the plant get nutrients.
especially phosphate ions and minerals, which can be hard for roots to access. And in return, the plant supplies the fungus with carbohydrates from photosynthesis. It's a partnership that's over 400 million years old.
And there are 2 main types to visualize, right? It's all about where the hyphae interact with the root cells.
Exactly. With ectomycorrhizal fungi, the hyphae form a dense sheath over the root surface. They go between the root cells, but they never actually enter them.
Okay, so they stay on the outside.
But with arbuscular mycorrhizal fungi, it's different. Their hyphae extend these specialized tree-like branching structures called arbuscules. And they
Fluoroseals.
go through the plant cell wall. They don't pierce the plasma membrane, but they push it inward, creating this intricate site for nutrient exchange deep inside the root cell.
So a much more intimate relationship.
A highly structured, intimate invasion for mutual benefit. Meanwhile, parasitic fungi use a similar tool, the haustoria, to just steal nutrients instead of exchanging them.
Okay, so that's structure. Let's pivot to reproduction. Fungi are famous for their spores.
And for good reason. They propagate mainly via these massive numbers of tiny, lightweight spores. They are the key to colonization. Wind, water, they get everywhere. A single puffball can release trillions.
Trillions. The numbers are astronomical now. The sexual life cycle it's complex. There's a strategic delay.
There is. It's predominantly haploid. Sexual reproduction kicks off when hyphae from two different mating types detect each other's pheromones.
And the first step is plasmogamy.
Plasmogamy, yes, the fusion of the cytoplasms from the two parent mycelia. But, and this is the critical part, the haploid nuclei often do not fuse right away.
This creates a unique, prolonged heterokaryotic stage,
Right.
or dikaryotic stage.
And you have to ask why. Why have two genetically distinct haploid nuclei just coexisting, dividing side by side, sometimes for months?
It must be a huge evolutionary advantage.
It's essentially genetic hedging. During that time, the fungus is out in the environment, competing, facing stress. If one of those two separate genomes has a good trait, or if the other has a flaw.
It's like having genetic insurance.
It is. You get the functional advantages of being deployed, two sets of instructions, without actually committing to the big genetic shuffle of meiosis just yet.
So the actual fusion of the nuclei, karyogamy, is delayed.
Delayed until the fungus is ready to produce its reproductive structure. That fusion produces the only diploid cell in the whole life cycle, and it's very brief.
And it's immediately followed by meiosis.
Right, which restores the haploid state and creates genetically diverse spores ready to go out and start a new, unique mycelium. It's a beautiful system.
But they don't always need sex, of course. Asexual reproduction is huge.
Rampant, especially among molds, the filamentous fungi that just produce haploid spores by mitosis, and yeasts, like Saccharomyces, which just bud off new cells.
And what about the ones that don't seem to have a sexual stage at all?
They were traditionally called Deuteromycetes, the imperfect fungi. But now, with modern genomic sequencing, we're constantly finding their sexual stages and reclassifying them.
That makes sense. Okay, so this complex life cycle must have taken eons to develop. Let's wind the clock back to their origins. They're in the opisthocons clade with animals.
Which is just remarkable. The name opisthocont refers to the single posterior flagellum in their ancestors. But here's the key finding. Fungi are more closely related to some unicellular protists called nuclearides, not to animals.
So multicellularity evolves completely separately in fungi and animals.
Independently, yes, which is a huge deal in evolutionary biology.
And that aquatic flagellated ancestor, it must have been a real pioneer.
One of the first. Fungi were likely colonizing land even before plants, maybe 505 million years ago, breaking down rock, creating soil.
Paving the way.
And we know those ancient mycorrhizal partnerships we talked about were established very early. We have fossils of arbuscule-like structures in plants from over 400 million years ago. This symbiosis is absolutely foundational to terrestrial life.
And the diversity that came from that is just stunning. We estimate, 2.2 to 3.8 million fungal species.
And we've only identified a tiny fraction of them. It's a vast, unexplored kingdom.
So let's try to group the major lineages, starting with the ones that still have links to that aquatic past.
Right, the basal groups. We have cryptomycetes and chytrids. Some cryptomycetes still have flagellated spores, reminding us of that primitive state. But chytrids are really defined by their unique flagellated spores called zoospores.
And chytrids they're infamous now.
They are, and it's an ecological tragedy. A chytrid fungus, Batrachochytrium, has been implicated in the global decline of some 500 amphibian species.
500.
It's devastating. And it shows how quickly a pathogen can disrupt ecosystems, especially when global trade helps it spread around the world.
So moving on to land, the fungi lose the flagellum.
And rely more on wind. Next, we get lineages defined by making a tough sexual structure, the zygosporangium.
That would be the Zupagomyces and Mucoromyces.
Right. Zupagomycetes are often parasites. You've probably heard of the fly death fungus, Entomophora. It manipulates the fly's brain, forcing it to climb up high before it dies to maximize spore dispersal.
That's incredible. And Mucoromycetes, that's black bread mold.
Exactly, like rhizopus. They usually have coenocytic hyphae, and their sexual reproduction ends in a tough multinucleate zygosporangium, a little survival pod.
And some of them have amazing dispersal tricks.
Oh, like Pilobolus. It can actually aim and shoot its sporangia up to 2.5 meters towards a light source. Just an astonishing feat. And crucially, the glomeromyces, the group that forms all the arbuscular mycorrhiza, are now classified here.
Okay, finally we get to the two big phyla that most people think of as fungi, the sac and club fungi.
The ones that really mastered that prolonged eukaryotic stage. First, the Ascomycetes or sac fungi.
They make their spores in a sac, an ascus.
Correct. Sexual spores, the ascospores, are made inside those asci, and those are often housed in a big fruiting body. The ascocarp, think of a truffle or a morel.
And their asexual spores are conidia.
Produced externally, yep. This group has huge economic importance.
And then the basidiomyces, the club fungi.
The classic mushrooms, puffballs, shelf fungi. Their defining structure is the club-shaped basidium. That's where karyogamy and meiosis happen to produce the basidiospores.
And the fruiting body is the basidiocarp, the mushroom we see.
Which is often just the visible part of a huge underground mycelium, sometimes growing in those fairy rings. And ecologically, they are critical. They are the world's best decomposers of lignin.
The tough stuff in wood.
The toughest stuff. Without them, the world would be buried in dead trees.
Which brings us right to their indispensable ecological role. Without fungi and bacteria, the planet would just grind to a halt.
That's not an exaggeration at all. They are the only organisms that can efficiently cycle key elements like carbon and nitrogen that are locked up in dead organic matter. Without them, ecosystems would just collapse.
We've covered mycorrhiza, but what about other mutualisms, like endophytes?
Right, endophytes. These are fungi, usually Ascomycetes, that live inside plant tissues without causing harm.
And they help the plant.
They can, significantly. Research on the cacao tree showed that endophytes act like an invisible shield, reducing leaf damage from pathogens.
And what about fungus-animal partnerships? The leafcutter ants are the classic example.
A classic for a reason. It's one of nature's great codependencies. The ants can't digest leaves, so they bring them back to their nest, chew them up, and use them to farm a specific type of fungus.
And the fungus is their food source.
Their only food source. They eat the protein-rich tips the fungus produces. It's a millions of years old agricultural system.
Then you have lichens, which aren't one organism at all.
No, they're a mashup, a symbiosis between a fungus, usually an ascomycete, and a photosynthetic partner algae or cyanobacteria.
The fungus provides the house.
And protection and water. The partner provides the food. They're the great pioneers, breaking down bare rock to create soil. They reproduce by sending out little packets of both partners, called soradia.
But as helpful as they are, about a third of fungi are parasites.
Yes, and they cause immense harm. Plant pathogens can wipe out 10 to 50% of a harvest. Chestnut blight is a famous tragic example.
And some are just toxic.
Very. The fungus Claviceps purpurea grows on rye and produces ergots. Consuming that grain historically led to a condition called ergotism.
What were the symptoms?
Horrific. Gangrene, muscle spasms, terrifying hallucinations. In fact, the raw material for the drug LSD is isolated from those same rye ergots.
And for humans, fungal infections or mycosis.
They range from common things like ringworm to really serious systemic infections. We already mentioned the chytrids in amphibians. For humans, opportunistic mycoses are a big threat.
Like yeast infections.
Right, Candida albicans is familiar. But there are much scarier emerging threats, like the multi-drug-resistant fungus C. auris, which is a serious global health concern.
Okay, so despite the dangers, we rely on them daily. Let's finish with all the good they do for us.
It is a vast list. I mean, beyond the amazing flavor of morels and truffles.
Whose scent mimics pig pheromones, right? To get them dug up.
Exactly, to aid spore dispersal. But we also use Saccharomyces yeast every day to make bread and beer.
And medicine. That history is just transformative.
Absolutely. The discovery of penicillin from the penicillium mold kicked off the antibiotic age. Fungi still give us crucial drugs like cholesterol-lowering statins and cyclosporine, the immune suppressant for organ transplants.
And they're still at the cutting edge of research.
They are. S. cerevisiae is a model organism helping us understand human diseases like Parkinson's. And there's even research into fungi like Gliocladium roseum, which produces hydrocarbons similar to diesel fuel.
Biofuel potential.
Huge biofuel potential. They really are the chemical engineers of the future.
What a dense and fascinating journey. So to recap, Fungi are master external absorbers with bodies made of chitin-reinforced hyphae.
And their life cycle strategically use that unique prolonged dikaryotic stage to maximize genetic variation.
And ultimately, they're the keystone recyclers that our ecosystems depend on. Ancient colonists whose partnerships literally built the world we know.
Even as they simultaneously hold the potential to cause catastrophic ecological collapse. It's a profound paradox.
So thinking back to those first moments of colonization, that bare land and the need for partnership, if fungi had failed to establish those mutualistic relationships with plants, the mycorrhizae, the lichens, what would the landscape of life on Earth even look like today?
It's almost impossible to imagine. The entire foundation of forests, of fields, it all rests on that ancient handshake. The world would be unrecognizable.
Indeed. Well, thank you for joining us for this deep dive into the fascinating world of fungi. We hope you feel significantly more well-informed.